Hello, guys. So today I will be solving the May 2019 Time Zone 1 IB past paper for biology. I scored a 7 on my May 2024 biology paper 1. So I'll be beginning on doing that. Private tutoring will be available soon. I'll be starting my own website where we tutor the full IB biology syllabus, and also private tutoring is available. And if you're interested in private tutoring, then please, my email will probably be in the description. So anyways, let us begin. So for the first question, which graph represents the change in cell surface area to volume ratio with increasing cell diameter? So we do have surface area in the y-axis and cell diameter in the x-axis. Now, we do know that as cell diameter increases, surface area decreases. Surface area to volume ratio, be careful, this is the surface area to volume ratio, right? So as surface area to volume ratio increases, what will happen is that the cell diameter, this as the cell diameter increases, the surface area decreases. Okay, the surface area to volume ratio decreases. And why is that? Because what will happen is that if the cell would not have enough space to burn things faster, so it's gonna take the process of burning foods and like spending energy is going to increase. Okay, so the time to like actually gain energy will increase, and burning energy will take place in a shorter period of time because the cell will need way more energy to function since it's a large cell, which is why cells end up dividing into two daughter cells. Therefore, the B is right, because as as the cell diameter increases, the surface area to volume ratio decreases, just because it's going to be way harder for the cell to actually function. It's going to take so much time, and the surface area to the volume ratio will begin to decrease. Okay, because for every surface area, there's going to be less volume. So this is it for the first question. So our answer is B in this case. Now for the second question, we have the image shows an electron micrograph of a part of a cell. Which features do two labeled structures X and Y have in common? So we have X, we have Y. So first, let's see what these two are. So one's the nucleus and one's the chloroplast, I'm guessing, because this is the nucleus, it has a double membrane, and it's really large in size compared to the other organelles. And this is the chloroplast because you can obviously see like all the chlorophyll inside it. So what do these two have in common? They are surrounded by a double membrane. This is technically true. You can even see the double membranes here. So this is true. They contain 70 S ribosomes. This is false. The chloroplast does contain 70 S ribosomes, but the nucleus contains 80 S ribosomes because the chloroplast was once a prokaryote. This is due to the anti-symbiotic theory. So it's a prokaryote, which is why it has 70 S ribosomes. Okay, so here endosymbiotic theory. This is just like a revision. They contain naked DNA. This is only for one of them, does not apply to both of them, because the prokaryotes, they contain naked DNA, which is in this case the chloroplast, and they are only found in leaf cells. This is false because the nucleus is not only found in leaf cells. So yeah, so the answer is A in this case. So for the next question, this image shows a sequence of changes in an organism. So here we have the changes. This looks like a penis. I'm guessing it is a penis. Oh no, sorry, this is a analysis of frog early development. Really my bad. Which is the change and which processes is it necessary to occur? So in this case, you have egg production. No, because it's not really the case. And the process necessary is not meiosis. No. Embryonic development and cell division, which is true because here we can see that specialization is actually taking place. So we can say that it's cell differentiation. So this does make sense, and this does make sense because it is the analysis of a frog. And C, excretion, no, because excretion is basically when you get the feces out. So this is definitely not excretion. And exocytosis, no, because exocytosis is basically a process where it comes to transportation. This has nothing to do with like transportation of molecules or whatnot. So no. Feeding, no. So we don't even have to talk about phagocytosis because feeding is wrong. So our only correct answer is B. This is a not this is an easy question. It's not a difficult question. Which process occurs by osmosis? Interesting question. Okay, so osmosis basically transport, a type of transport. It does not go up a concentration gradient. Okay, so what happens in osmosis? Let's see, uptake of water by cells in the wall of the intestine. Yes, that can happen. That these processes do occur by osmosis because in this question, they are asking us process occur by osmosis. So like any process that basically osmosis can take place. Loss of water from a plant cell in a hypertonic environment, this too can happen. For example, the plant can be in soil and the soil is really salty. So the plant ends up losing water due to the salty soil, okay? And then you have evaporation of water from sweat on the skin surface. No, that does not have osmosis take place. And this actually needs a really, really, really high amount of energy in order for the hydrogen bonds to break down. So no, so it's just one and two. So let's look for the options that give us one and two. So yes, the answer is B. Okay, so table 5 shows us the number of cells in various stages of the cycle in 4 samples of ovarian tissues from different patients. So we have which sample A, B, C that has the highest mitotic index. So let us calculate the mitotic index. So firstly, how do you calculate it? Mitotic index is basically prophase plus metaphase plus anaphase plus telophase divided by interphase plus prophase plus metaphase plus anaphase plus telophase. Okay, this is basically it. So we technically have to add each one, but this is really easy. I'm just gonna do it really quick in another color. So we have 1, 2, 3, 4, right? Um, so we have 4 over 50. Okay, in this case, they even gave us total, so we didn't have to like do all the work. 4 over 50. And here, keep in mind, you're not allowed to use a calculator. Then here you have 1, 2, 4. You also have 4. So you have 4 over 100. And then here you have 4 over 25. And then here you have 3 over 75. And basically that's it. They made it this simple for you. So we do know that in this case, the largest number out of all of these is in fact 4 over 25, right? So in this case, our answer is C. Okay. So now what type of molecule is formed by the chemical reaction shown in the diagram? So here we can directly spot the H2O as one of the products, right? So now we know that this is a condensation reaction, okay? But how can we know what the actual molecule is, right? So we can actually see 2 glucose molecules. So this is glucose and this is glucose because remember that glucose has 6 sugars. So in this case, we do have we do actually have monosaccharide, monosaccharide. So here we end up getting a disaccharide through a condensation reaction. Okay. Which substance must be transported in the blood by lipoprotein complexes? Well, this is quite easy. We know that it's cholesterol, okay? Because oxygen does not need to be transported by lipoprotein complexes, no. And amino acids and sodium chloride are transported in different ways. So yeah. What is found in insulin molecules? Phosphate, no. Nucleotides, no. Peptide bonds, yes. And glycerol, no. So we have our answers, peptide bonds. This was quite easy. Now for the question, the graph shows the results of an investigation into the activity of turnip peroxidase. Okay. So this is our enzyme, I guess. The accumulation of the product of the reaction catalyzed by the enzyme is shown at different pH values. So we do know that enzymes, they're affected by pH, by temperature as well. Okay. So here we, so between the data and the graph, what is probably the most optimum pH for ternary peroxidase? Okay, so keep in mind that you have to know that the optimum pH is basically when the enzyme works at its best, when it's working at its highest rate of reaction, when the reaction is taking the least time possible, right? And after that, like if you surpass that certain optimum pH or temperature, whatever it is, then the enzyme may end up denaturing, and the denaturation is irreversible. So in this case, we actually need to look at each answer. So between 3 and 5, definitely not, because the concentration of the product is way, way, way less than what it could possibly be, which is any of these. Between 10 and 11, so we really cannot even see 11, but we do have 10, and it does not, it isn't as high as possible, like it's not working at such a high time. And yeah, basically, so no, 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 no. Between 7 and 8, which is probably here, um it is possibly 7, 8, because 7 is the highest, so this may be. And between 9 and 10, no, because we have 7 that's highest, there's the most concentration of product. So it is 7 and 8. C is our answer. For 10, the gene that codes for a particular polypeptide includes the base sequence shown. Okay. So here, we have to actually, what is the best sequence of the mRNA molecule which is complementary to this base? So what we end up doing is just, we just end up translating this. So we have, because G turns into C and A turns into U, and oh, sorry, I wrote C, but it's U. A turns into U, and then T turns into A. Okay, so I think I like kind of said them all, I guess. But basically, we have C, we have U again, then we have C again, and then we have A, and then we have U, and then we have wait, so C turns to G, right? Yeah, so G, 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 T. So I guess that if the answer is, I'm just looking at the answers, guys. So it's not this one. It's not this one because of the T. So the T wasn't changed. It's not this one. So I'm guessing, sorry, so it's not this one. So I'm guessing it's D. Yeah, we got the exact, oh, I forgot changes to A. Okay. So I think that's the exact answer that we got. Okay, so now we have 11. So, okay, so this needs quite some reading. Yeast cells were incubated with and without glucose, with and without, very important, at 3 different temperatures for a period of time of 4 minutes, during which the rate of CO2 production was measured with a CO2 sensor. Okay, so we have with glucose and without glucose is very important, and we have the temperature. Um, okay, so rate of CO2 production. What conclusion can be drawn from the results of the experiment? So, yeast uses lipids rather than glucose in respirations at low temperature. Um, Not necessarily, no. I'm gonna read the question again just to really focus on what they want. So, okay. So it's not A. Addition of glucose has greater impact on rates of cell respiration at lower temperatures. Not necessarily, because at 4 it didn't really do much. At 30 it kind of, but that doesn't, not necessarily, because as the temperature increased, the glucose increased. I mean the ones with glucose increased, so that's false because you can see that here. Rate of cell respiration increases with temperature, that is true. Um, because we can just tell that they increase as the temperature increases in all, with and without glucose, so that is true. More glucose is produced at higher temperature, so not really, that's not the answer, because they're talking about the like actual rate of CO2 production, so no, so it's C. 12. The graph shows the effect of increasing light intensity on the rate of CO2 uptake by the species of green plant, maintaining conditions consistent temperature and CO2 concentration, or constant temperature and CO2 concentration. Okay, which statement is consistent with the graph? So they just began by just like showing us the graph and describing it. We have, this is our y-axis, this is our x-axis. So here they just say photosynthesis stops at high light intensity. So technically photosynthesis does not stop, but what happens that there's enough light, like it's saturated, that's why it forms into a plateau, which is not that it stops, but just the rate can't increase. So this is false. It's not that it stops. Rate of photosynthesis increases with temperature. So this is true to a certain extent, but this is not answering the question, because this is not, like I said, this, it has to be consistent with the graph. B is not consistent with the graph, okay? Okay, and remember there are three things that affect the photosynthesis. So three things. So temperature, CO2 concentration, and light intensity, okay? Cell respiration leads to net production of CO2 at low light intensity. So that leads to net production of CO2 at low light. To a certain extent, yes, it does, just because here, like the rate of CO2 uptake isn't at a high like light intensity, so it is C. Um, and there is a negative correlation between CO2 uptake and light intensity. This is not true, because as you can see, it keeps increasing and it doesn't change. So it cannot possibly be negative. So we say that cell respiration leads to net production of CO2 at low light intensity, because net production of CO2 is just because the rate of CO2 uptake can't increase at the low light intensity anymore, because it's net, it's full, it cannot increase. So now for question 13. The system of sex determination in chimpanzees is the same as in other mammals. A chimpanzee has 48 chromosomes in the nuclei of its body cells. Okay, so body cells. Here they mean somatic cells. Okay. Um, So, and we have 48 chromosomes and then the nuclei, which is technically a somatic cell. What can be deduced from this information? Not the chimpanzees, because all chimpanzees have the same chromosomes, so we can't know the sex chromosomes. It did not mention anything about the sex chromosomes. The number of genes in each organism, in each chromosome, no. This is not something we can possibly know. Whether disjunction has occurred, but this has to do with Down syndrome, or in other words, trisomy 21. The number of autosomes in a diploid cell, yes, this is exactly what they told us, because they told us in a somatic cell, which is a diploid cell, okay, 2n. So yeah, and they told us it has 48 chromosomes. So what can be deduced from this information? This is a very, very, very clear-cut answer. It's very straightforward for the student to figure out. At which stage of meiosis are bivalents formed? So we know that bivalence is basically when, okay, this is not how you draw a chromosome. You can, but I personally don't draw it like that. I just draw it like that. Something along this way. And I'm gonna like use another color so you guys can clearly see. So bivalents are kind of when, no, okay guys, I'm trying to really draw, so excuse my drawing skills. So bivalence is something like that, when they just cross over, I guess. So it's just something like that. Okay, so ignore my very, very ugly chromosome, guys. I'm just really trying. Okay, so it's just really something like that, guys. Okay, so it's basically the crossing over. And this technically happens in meiosis. Okay, so they didn't try to trick us. They directly told us it takes place in meiosis. But when in meiosis, it actually takes place in the first stage of meiosis, meiosis 1. Okay. So it's better to just write them in normal numerals, and this takes place in prophase 1. Okay. Creeper chickens, creeper in chickens is a condition in which the chickens are born with very short legs. The creeper C allele is dominant. Okay, dominant. Okay, and they also gave us this. Embryos which are homozygous in the dominant allele fail to develop into viable chickens and die before they hatch. Die before they hatch. Okay. When they give you a really long paragraph like that, guys, just make sure you read it all in order to get all of your information, annotate, because really it's also about the grammar, like I emphasize in all of my videos. Um, what phenotypic ratio would you expect in the live offspring of a cross between two creeper chickens? Exactly. So two creeper chickens. So creeper chickens is basically when they are heterozygous, right? Okay. So okay, so this is actually not a straightforward question. So creeper chicken, what is it? It's when they are homozygous, heterozygous, so sorry for the confusion, guys. It means they have one dominant allele, which is the creeper chicken allele, and one recessive allele, which is the normal allele. So this is how they are creeper chickens. But if you get two genes of creeper, then this is homozygous dominant, and this will lead to death. Okay, now they're asking us the phenotypic ratio for live offspring between two creeper chickens. So we know that since they are both creeper chickens, they both have a heterozygous genotype, right? So we're gonna get, so these three, what phenotypic ratio would you expect in the live offspring of a cross between two creeper chickens? So here we cannot talk about this one. So we have to talk about these three because all of these will be alive, right? But since it's dominant, these two will be creeper. So creeper, and this would be normal. So we have a two creeper, one normal, or like basically that's how it works, I guess. Or like 50% creeper, 25% normal. Like you can just say these. But anyway, so let's check our answer. So it's not all creeper, no. One creeper, two normal, no, it's the opposite. Two creeper, one normal, yes. So our answer is C. So now for question 16, which pedigree chart is consistent with the inheritance of red-green color blindness? So basically, we need to actually look at this in great detail. So here we have um all like the key, so normal vision female and color blind male, for example, right? So in this case, we need to look at each one. So for A, we can see that both the parents, they're normal, and then we have both the males having this the red green color blindness, and this is because it's actually a recessive disease, which is X-linked. So, which this does work because if the mother has it, then she can just give one of her X's. So basically, she's like X normal, X like colorblind. I guess I'm just giving you anything, but this like they probably got this X. The girl is probably heterozygous, or she doesn't have it, or she carries it, and yeah, so this is it. So I guess it's A because it does work with color blindness. I'm checking if B works for color blindness. Um, so B cannot work for color blindness just because it is recessive. So if the mother had it and the boy had it, the son had it, then the two other sons should have it. Okay. So basically this is it. And the mother and daughter can't both have it in this case. Okay, so this is basically it because if the mother would be homozygous, right? So yeah, so her sons would all have to have it, and the girl can't have it if her dad doesn't have it because the dad would directly show because he has one X. So I don't know if you understood this. Basically, the dad has XY, and this X has to be normal because if it wasn't normal, then he'd automatically be colorblind because he has one X chromosome, right? So his daughter would automatically have it if the mother has it. You got it? Because her mother would be XN. I mean, XN, XN. She'd get one from the mother and one from the father if he had it. So she can't possibly get it because it doesn't work that way. And then in this case, this is just also not working because the daughter need the dad if the dad the daughter needs to get it from the dad as well. And the dad would show if he had it. The dad can't be a carrier because he has only one X chromosome. You got me? So he's X normal and Y. So he would end up giving his daughter an X normal and she wouldn't have colorblind disease, so she can't. So now for question 17, what organism can be best described as a saprotroph? So a saprotroph, so I'm going to give you like a small lesson now. Saprotroph is basically a heterotroph. So heterotrophs do not make their own nutrients. They actually eat other organisms. They digest other organisms in order to get their fair share of nutrients, right? And they do it by enzymes, external enzymes that are located in the gut. And this is basically how they do it. And they are actually the like decomposers of like the ecosystem. So the ecosystem couldn't really do it without them. So what organism is best described as a saprotroph? A fungus that digests its food externally, this is true, absorbs the products of digestion. Yes, this is true. A beetle that feeds by ingesting, this is not true. It does not ingest, it digests the dung of other animal species and digests its food internally. No, it digests actually externally. So this is false. A single-celled eukaryote that is able to photosynthesize and consume smaller organisms by endocytosis. So basically, this is full of really specific information, like eukaryote photosynthesize smaller organisms by endocytosis. First, it does not photosynthesize because it's a heterotroph, so it actually, like I previously mentioned, digests its organisms from other organisms in order to have its nutrients. It does not make its own nutrients. Photosynthesis is basically the process of creating your nutrients. Okay, so this is false. A giraffe that feeds by ingesting leaves from an acacia tree. So no, because this does not require enzymes and does not ingest, it digests. So this is false. So our answer is A. Now for 18. So I'm going to use another color. The diagram shows the food web for an aquatic ecosystem in which letters R, Z represent individual species. Which organism is a tertiary consumer? So here we have, like, I think these are like the autotrophs. Yeah. But anyway, so I think these are the autotrophs. I think this is the primary consumer. This is the secondary consumer. And these are also secondary consumers. So this is a secondary, oh no, this is a secondary, no, sorry. This is also a secondary consumer, I think. Yeah. And this is a tertiary consumer. Okay, so, and these are just producers. So if you want this to make you easier, your life easier, so you have producer, primary, secondary, and so forth. I think you got, and then you have quaternary in the end as well. But I think you guys ended up getting the process. So for the answer for 18, it is organism U. Okay, and organism Z isn't any of the options, so we can't even consider that. Consider that. So question 19, which organisms produce methane in anaerobic environments such as waterlogged soils? So this is a quite easy one, so we know that it's archaea. Okay, because eukaryotes do not do this. They do not produce methane, nor do bacteria or fungi. 20. Which are examples of homologous structures? So basically, what are homologous structures? They're basically the opposite of analogous structures. So they're basically like kind of, in a way, I'm trying to find a good way to explain it, but they're basically the same features that are, or different features that are used in the same way. So the wings of bats and butterflies, they're not homologous just because They are, because homologous structure is when they're actually like kind of related. So the wings of bats and butterflies, no, just because they use them in the same way they use them to fly. So this is not the answer. The fins of fish and whales, no. The hind limbs of frogs and grasshoppers. And we have the four limbs of primates and penguins, which is basically our answers. Okay. For question 21, what's an example of natural selection? So natural selection, I've said it in many of my other videos, but basically it's survival of the fittest. So basically, it's when the organism with the features that help us, that help it best adapt to the environment surrounding it. And this is how natural selection takes place. It's because nature selects it, because its features are most likely aligned with nature, so it has the most advantages. So let's look at our answers. A giraffe stretching its neck to reach higher leaves, no. A juvenile bird learning how to sing, no. This does not help its survival, so this is no. Development of antibiotic resistance in bacteria, yes, because this does help its survival. And selective breeding of tail cats, no. So the answer is C. 22. What feature does this organism have in common with all members of the phylum Chordata? So let's look at this one. So because its middle is the same. So if you literally split it in half, it'll look the exact same. It has bilateral symmetry, just like the phylum Chordata. Okay. So yeah, because the phylum Chordata does not have to have a chitinous exoskeleton, for example. So 23. Um, so the DNA base logs in a gene coding for a particular protein in 4 different species are outlined are shown, sorry. Locations where mutations have occurred, resulting in changes to the base sequences are outlined in boxes. Okay, so now we actually need to look at this and see how it is. So we need to actually see how similar they are. So let's look at their similarities. So we can see that species 2 and 3 are the most similar. We can see all the similarities here. We can see that that species 1 is really, really different. So we can see from here that like these two, they're different here. Both ways are both different. But here this one's similar. This one's not. And we can see that there's some similarities here. But here there's no similarities. So these are the two most similar. These are quite similar, and this is the last one. So our answer is going to be, let's just look at the answers. 3 and 4 cannot be this close to each other. They cannot be in this clade. So the name is clade, guys. Keep in mind, these are clades. Clades. Okay. So no, because 4 and 1. So we're just looking at the specific clades. It saves us time. 1 and 2, no. 2 and 3, yes. This is our answer. Where in the human body is lipase produced? So this is not elimination, because you actually should know this. It is in the pancreas. So here we just look at our answers. Not in the stomach. It is in the pancreas, not gallbladder, not liver. That's it. This is simple. So what are these structures? So let us read. Now we have to have a good hint, is that the IB exams, they're like they just follow section by section. They start off by 1.1, they end by 6.6 or 6.5, or wherever it is. So just if you're confused or you don't know what to focus on, just remember the section that the chapter is in. I used to do that a lot, used to help a lot. What are these structures? Fatty acids in the small intestine, no, because they don't have this shape, they don't have the structure, no, because these are like kind of spiky a bit. They're pointy. Bacteria in the large intestine, no. Villi in the small intestine, yes, just because it helps absorption. Okay. And then you have feces ingested from the large intestine. No, these are not feces. This is false. okay, because they have a size for a reason and they're pointy this way, and the feces can't be this much in size. So which feature of capillaries distinguishes them from arteries and veins? Narrow. Okay, so capillaries versus arteries and veins, right? So narrow diameter, which is true because arteries and veins do not have narrow diameters, whereas capillaries can. With it, therefore, they can deliver the product. Valves to prevent backflow, thick muscular walls, and elastic tissue. They do have valves to prevent backflow, so it is narrow diameter. Now, for question 27, the diagram shows major events involved in the formation of a blood clot. Okay, so we need to look at this in detail. Um, so yeah, we need to look at this in detail. So tissue damage, clotting from platelets, damage cells, plasma, and enzymic cascade, factor W, factor X, factor Y. So what is factor Y? Factor Y is fibrinogen. Okay, in this case it is. 28, which statement applies to HIV? HIV infects red blood cells from resulting in decreased population of hemoglobin. No, it basically it decreases phagocytes. HIV can be effectively treated using antibiotics, not necessarily effectively treated. So no, it cannot be technically treated. HIV can only be transmitted by sexual intercourse. No, also by blood transfusion and also by breastfeeding in many other ways. So this is false. HIV causes a reduction in production of antibodies. Yes. Okay, and in the production of phagocytes. This diagram shows a graph of action potential. So what's happening at X? At X, the sodium channels are closing just because it's between depolarization and repolarization. So the sodium channels end up closing. And for the last question, the diagram shows the human female reproductive system. So here our answer is what is produced by structure X, estrogen, and progesterone. So this is it. Thank you so much, guys, for watching. If you want private tutoring, check my email in my link below. And my classes will be coming soon. Bye.